I am happy to introduce today our lecturer from Berkeley University, Adrisha Sarkar, who is our long-term collaborator from Ashoka Joy Group and with whom we had pleasure to meet personally several times. So let me introduce a short bio for Adrisha. She is a PhD candidate and graduate student researcher in the Applied Science and Technology program at UC Berkeley, where she also completed her undergraduate studies. Adrisha's research focuses on leveraging quantum sensing and nanotechnology to address complex biological and chemical challenges. And Adrisha's topic today is quantum sensing applications of nanodiamonds in microdroplets. Adrisha, the stage is yours.
Thank you so much, Olga. I'm excited to be presenting here today. And so in today's talk, I will be covering some of our recently published work in which we demonstrate the integration of two powerful technologies, droplet microfluidics with nanodiamond quantum sensing. Combining these two technologies really has helped us set up a platform for practical biological and chemical measurements. Kind of some of the key innovations I would like to highlight today is how we implement a diamond-based measurement in microdroplets which are flowing to develop a rapid biological or chemical analyte detection with good sensitivity. And so I will walk through some of our findings in our publication, as well as some of the upcoming directions we are pursuing in the lab. So I'd like to start with something we all became very familiar with during the COVID pandemic, which is this COVID test, which is essentially a lateral flow assay. And we saw how sensitive and rapid detection of biological analytes can be very, very impactful and important. Work done by Miller and collaborators showed that replacing the gold nanoparticles in these lateral flow assays with fluorescent nanodiamond can be very impactful in terms of improving the sensitivity of the lateral flow assay. And they really improve sensitivity by many orders of magnitude. And so we would like to build on this work and integrate fluorescent nanodiamond particles with a variety of kind of biological samples and apply this sensing to many more biological samples. Secondly, a terrific measurement that is kind of typically done in the field uses this bulk single crystal diamond with surface NVs interrogating a sample kind of closely sitting on top of the bulk diamond. So here we are able to accomplish very sensitive measurements, but possibly at times the biological sample handling can be tricky. We have a bulk sample residing above the diamond chip, but we're only able to interrogate to some level within this total sample. And so building off of this measurement, we would like to re-envision this geometry to better handle biological samples and integrate fluorescent nanodiamond. So with these two foundational measurements in mind, we choose the approach of introducing nanodiamond particles into microdroplets to be able to interact the sensor with biological and chemical analytes of interest. So you can think of the droplet as a microconfinement where we can control the interaction of diamond particles with biological and chemical analytes. So we can also measure multiple of these droplets and putting all of this together, it really lends towards setting up a potential measurement that can have good sensitivity as well as high throughput. So a typical droplet microfluidic experiment looks something like this, where we have an array of droplets being generated relatively fast. Each of these droplets are highly monodispersed and essentially identical to each other. And we can think of each of these droplets as a microbeaker and a picovolume of fluid. Using this technology, applications like reaction kinetics measurements, single-cell analysis, and flow cytometry-like measurements have been accomplished throughout the microfluidic field. And by integrating nanodiamond particles into these systems, we can then use nanodiamond to interrogate these types of experiments. More specifically, in our system, by integrating diamond particles into microfluidics, we can monitor chemical reactions, measure biological assays, measure free radicals produced by living cells in droplets, possibly develop some sort of portable diagnostics, and use droplets for material sorting as well. So keeping kind of this wide variety of experiments in mind, I would like to tell you about how we develop a measurement in droplets using nanodiamonds. So we generate these microdroplets using devices that we design and fabricate ourselves. We use a soft lithography process to etch channels into PDMS, and this PDMS is then bonded to a glass cover slip. In these hollow channels, we can connect them to fluid reservoirs and drive fluid through the channels to generate droplets. And we also can interrogate these droplets using an inverted wide field microscope setup. So here at the kind of center of this schematic, we have droplets flowing by a laser spot where we can image and detect droplets. Below this detection region, we also have a microwave loop antenna to apply microwaves for NV center protocols. And during all of this, we're able to image using this wide field microscope. Now, as you can see here, there are kind of various complex features along the microfluidic channel. And so we use these to create the microdroplets. So to start, at the first junction, we have an aqueous phase pinched by an oil phase to generate droplets. Subsequently, these droplets encounter a second junction where we're able to inject oil between droplets and increase the interdroplet spacing. Downstream of these two junctions, we can measure droplets at the laser spot. So in the first two junctions, we create the droplets, modulate the droplet spacing, and then all of these properties are kind of maintained over a long distance throughout the droplet, and this is what a real-time experiment looks like. So the key features to kind of note here are at the first junction, we set droplet size. At the second junction, we modulate the droplet spacing, and all of those properties are maintained throughout the chip downstream of these junctions. Furthermore, to have real-time control of droplet size, we use a program that we have developed, which has a feedback loop that is based on measuring the droplet diameters. So in this video, as you can see, these green rings are detecting droplet sizes in real time, and a feedback loop is used to regulate fluidic pressures to change the droplet size on command. So you can see kind of, as I scrub through this video, we step up the droplet size to larger and larger sizes using this program. And kind of key feature here is that while all of this droplet microfluidics is very stable, this program kind of makes things much more user-friendly, as well as if there's some drift in the droplet size or change in the droplet size, this program can be used to correct for that and check that the droplet size is well maintained throughout the experiment. So here is kind of a graph representation of the radius of a droplet being changed over time. Now, within these microdroplets, we can also encapsulate nanodiamonds. So we do this just by adding diamond to the aqueous flow. And in the image on the left here, we're looking at stationary droplets, and these are fluorescent images of those droplets. These nanometer-sized particles are well suspended throughout the droplet, so the fluorescence through the droplet looks very even. For these micron-sized particles, when the droplet is stationary, since these 3-micron-sized particles are heavy, they do sink to the bottom of the droplet, and that's what kind of creates this dark halo within the droplet. So we have control over how we're loading different diamond particle sizes within the droplet. We can also control the number of diamond particles per droplet. So here we have a single diamond particle encapsulated in a droplet, and really we can span this 1:1 or many:1 encapsulation regime. Now, if we look at one of these stationary microdroplets in real time, you can see that the particles are undergoing motion. So particles within the droplet undergo Brownian motion even within a stationary droplet, and we can track and characterize this Brownian motion. So this is an example of a few Brownian motion tracks, and the key point here is that these 100 nanometer particles really sample up to a 10 micrometer sized space within the droplet. And so there's a lot of mixing and interaction between diamond particles and the analyte within the droplet. Before I delve into the measurements that we made, I want to just provide a brief overview of what optically detected magnetic resonance is and how we are using it. So, as we all know, we can polarize the NV center with green laser light, and the fluorescence emitted by the NV center is spin state dependent. We can use on-resonant microwaves to go from bright to a slightly dimmer spin state. A typical ODMR measurement uses a continuous wave laser while simultaneously applying microwaves and counting the photons emitted. So an example of an ODMR spectrum is in this bottom right corner. So you can see this characteristic dip at the microwave resonance condition. This is typically referred to as ODMR contrast and often represented as a percentage. We can use ODMR contrast as a sensor for chemical analyte concentrations. So by this, I kind of mean that ODMR is an indirect measurement of T1, and any dipolar interactions with the NV center that cause relaxation will perturb this contrast value, and we can use that to infer concentrations of chemical analytes like paramagnetic ions. And so with this, we measure ODMR contrast in a stationary microdroplet with fluorescent nanodiamond. Here, we measure the PL as we sweep across a range of microwave frequencies. And as you can see, the signal is quite noisy. A typical strategy used in these kind of measurements involves using a lock-in amplifier to better resolve this signal. So here in this graph, at each microwave frequency, we're modulating the microwave on and off and locking into the fluorescence signal that is modulated at that same frequency to generate this ODMR curve. Here we are able to measure contrast much better. So with this better SNR, we can use this contrast value over here to now think about sensing a chemical analyte. So I'd like to highlight that usually this contrast value is around a percent signal, so 1%, and we want to see small changes on this signal to be able to infer chemical analyte concentrations. So here we are measuring contrast at this resonance condition over time. And so we measure this C-lock value normalized to the total droplet fluorescence and plot contrast over 90 seconds. The percent error in this measurement is relatively high, 13%. And with this kind of resolution of contrast, we are not very well set up for being able to sense small changes in contrast values. So really, that is a challenge here where we would like to measure contrast with more SNR so that we can use it to sense chemical analytes. And also a key idea to remember here is that we need to be able to have a normalization strategy where we are able to compare measurements across different droplets. So to figure out a lot of this puzzle, we relied on some of the microfluidics that we are able to develop. So we implement a double lock-in strategy to be able to improve on this measurement. So as you may recall, I mentioned how we have control over droplet size, interdroplet spacing, and this is maintained throughout the chip. So now if we kind of visualize what the fluorescence at the detection region of the chip looks like, it looks something like this, where we have a very periodic oscillation of the microdroplets through the field of detection. And we can plot the fluorescence of droplets flowing by this field of detection, and it looks something like this, where we have these sine wave-like oscillations where the peaks are when the droplet is in the field of view, and the troughs are the droplet spacing, essentially. And so we can do this stably from about 2 Hertz to 350 Hertz, and really have a sine wave-like modulation from flow for the microdroplets. We can use this idea to now measure the total fluorescence of a droplet with some sort of locked-in strategy. And so now we can measure the microwave signal normalized to a locked-in measurement of the modulated droplet flow. And we use this kind of ratiometric definition of contrast for better SNR measurement of contrast in flowing microdroplets. So to summarize all of the pieces of the puzzle together, this is what our experiment protocol looks like. We have a continuous wave laser, which is applied to the sample. We are also applying modulated microwaves at about a 1 kilohertz frequency. The flow of droplets is modulated less than 300 hertz. And all of this information is then encoded in the fluorescent signal that we measure and the PL readout. So the data looks something like this, where we have droplets flowing and this sine wave-like modulation from the droplet flow. And you can see how in the 1st second, the 50th second, and 500th second, everything remains stable, which you can see visually. If we look at a zoomed in section of this, as each of these droplets flow through the field of view, we can see oscillations based on the microwave modulation. And so I'd just like to reiterate that we need to measure this contrast value and also measure small changes on this to be able to measure an analyte. So with this data and strategy, we can also look at this time domain data in the frequency domain, and we get some interesting peaks. So at the low frequency range, we have a droplet frequency peak and some side harmonics based on how square wave or sine wave-like the modulation is. And at the higher frequency, we have the microwave modulated peak at the kilohertz, at 1 kilohertz, and two sidebands that show up because of the amplitude modulation created by the microdroplets flowing. We can look at these frequency peaks across time in this spectrogram. So essentially, we can see that these peaks are stable over an hour through this spectrogram analysis. With this information, we can normalize the microwave information to the total droplet fluorescence and create this double locked-in contrast measurement. And indeed, it is an improvement. The percent error over here is now 2%. And so we are much better positioned to use contrast to sense an analyte. And over here, we are generating about 300,000 droplets over 2.5 hours. So really, we are doing this measurement with high throughput. We can use Allen deviation to characterize systematic error and long time drift in our setup. So essentially, the key takeaway here is that our measurement scales with time. And so not only can we do kind of single shot or short time measurements, but we can also improve measurement sensitivity by increasing averaging. So we're able to average for up to an hour and improve the percent error by 30-fold. We also characterized how kind of innate features of the experiment do not lead to a change in contrast values. So here we measure the change in contrast based on changes in nanodiamond concentrations as well as laser power changes. In the graph on the left over here, we increase nanodiamond concentration by 500% and see roughly a 10% change in the contrast measured. Typically, the variation in nanodiamond loading between droplets is something less than 0.3%. And so whenever we measure contrast in these measurements, we can assume that the changes in contrast are not from changes in the number of nanodiamond encapsulated within a droplet. So we also measure contrast as a function of laser power at a fixed microwave power. So here we typically operate in this 100 milliwatt range, and our laser is characterized with about a 1% fluctuation over time. And you can really see in this inset that some 1% fluctuation in laser power would not lead to a measurable change in contrast. And so really kind of putting together the microfluidics, the double lock-in measurement, and characterizing the stability of our measurement sets us up to now measure a chemical analyte of interest. So we measured paramagnetic ions in droplets. We started by measuring gadolinium ions in a microdroplet. So in this first graph over here, we generate a calibration curve from 500 nanomolar to 20 micromolar. And we're able to measure down to 100 nanomolar sensitivity for gadolinium. Some kind of interesting features of this calibration curve are the time it takes to generate this. People who work in this space might know that generating these calibration curves can often be time-consuming or tedious. And so each of these data points that we collect are for less than 2 minutes. And we use a valve to switch between these samples, and that switching is also fast, so we can generate this data quickly. So I have a little video to kind of show how a sample is switched on chip. So we start with these red droplets and then move to a spacer fluid in between the two samples. And then now you can see blue droplets have entered the chip, and so we have switched from one sample to the spacer fluid and the next sample within less than a minute timescale. And so really generating this data can be done with good sensitivity and quite quickly. Gadolinium is a high-spin molecule, and really eventually we want to measure more biologically relevant analytes like reactive oxygen species. So we measured a stable radical tempol, and we are able to hit about a 2 micromolar sensitivity with this measurement. So really to kind of characterize the experiments that we have done so far, we're able to use low volumes and detect low concentrations of gadolinium and tempol. And an interesting kind of fact within this experiment is that we use a small volume of diamond particles, which relates to kind of almost a $0.50 cost over an hour. And so we can look at this diagram on the left here to kind of characterize our sensitivity. So in this plot, the ideal position would be to be in this bottom left corner where we're sensing low analyte concentrations at low sample volumes. And our work kind of falls in this orange region where we are able to characterize this 2 micromolar tempol and 100 nanomolar gadolinium. For samples where we have access to more sample volume, we can use averaging to improve sensitivity further at the cost of sample volume. And so kind of all of these experiments so far have helped start a measurement using diamond within droplets and characterize all of the features of this experiment well. But there are many applications that we are working towards improving with this setup. So to kind of briefly introduce a flavor of some of these experiments, we can use these droplet generators to mix two reagents together and initiate a reaction. We can also encapsulate diamond within these droplets and then use this diamond to kind of measure some sort of product buildup or rates of reaction. We can also sort materials using droplet microfluidics. So you could imagine as these droplets flow, we use an electric field to select for and throw away droplets based on a signal measured using diamond. We're also interested in developing biological assays within these droplets, and this can be done by kind of integrating antibody targeting to proteins within this droplet. So some of the initial strategies that we are working with involve adding polystyrene beads into the droplet, as well as a target protein. So now the droplet hosts a target protein that we are trying to measure, and diamond, as well as beads that have antibodies targeted to this protein. And so we can think towards developing an ELISA-like assay within the droplet, where we develop this kind of sandwich where we have a bead, protein, and then nanodiamond, and we can measure the buildup of diamond around a bead to be able to infer protein concentrations. And this would be a very impactful measurement for biological assays. We're also very interested in measuring free radicals and doing this sensing in living cells and living systems. So some work in this area we have done kind of involves various cell types to be able to set up a diamond-based measurement in these microdroplets. So in the image in the top left corner over here, the bright field image shows the morphology of a yeast cell. And so essentially in this microdroplet, we have encapsulated nanodiamond as well as yeast cells. And in the fluorescence image, you can see the nanodiamond as well. So now we are able to kind of add cells to these microdroplets. And you could imagine introducing a buffer inside this droplet that would lyse the cellular material throughout the droplet, and we can sense cellular materials lysed in the droplet using diamond. We could also sense cellular secretions within the droplet or work towards integrating some form of uptake of diamond into cells and measuring that within droplets. So really we can add in biological cell samples into these droplets and now probe those cells with diamond in various ways. We have also been working with root samples. So here we grow plant roots in a microfluidics chip and inoculate them with diamond. So here you can see a plant growing in a soil-like matrix in the microfluidics chip. And we can expose different portions of this root to diamond, and the plant root is able to uptake some of this diamond, and so we can probe the rhizosphere of a systemic root. On the right here, we have a confocal scan of the root hair. And so essentially, the red fluorescence we see is partially due to autofluorescence, but also due to diamond particle uptake. And we can use ODMR-like measurements across root hairs to measure paramagnetic ions that reside in the rhizosphere. Lastly, I think most of the techniques that we have developed so far rely on simple wide field optics, as well as miniaturizable microfluidics, and the cost of diamond in this experiment is quite low. So it does lend towards a possible commercialization of a portable device and make this technology really accessible to various people working with biological and chemical samples. So thank you for listening to my talk, and please do read our recently published paper for more details about the experiment, and all of the experiments elucidated in this presentation could not have been possible without my collaborators in our group, Zach Jones, Madhur Parashar, Brooke Newell, Emmanuel Druga, and my mentor and PI, Ashoka Joy. So yeah, thank you for listening, and I'm happy to take any questions.
Adrisha, thank you so much for your excellent presentation. It's very exciting to see a big progress over just few years in a new field for you and Ashok's group that was started in Ashok's lab. So we are open for questions from the audience. I have, please.
Oh yeah, hi, Adrisha. Yeah, a couple maybe questions. If you go to the gadolinium like calibration curve, there was like this little bump, I think, at one concentration. Do you know why that might be, or if it's just like noise or something? And then I guess the second question would be, how do you see getting this noise level down even further than like the 2% kind of variation that you saw with the double lock-in?
Yeah. So I guess you're talking about this bump over here.
Yeah, yeah.
Yeah, so I don't have a good answer for why we see this bump, and I think that all of these measurements are statistically significant, and this is just what the calibration curve looks like. But in terms of your question about how can we improve this measurement further, I do think there's still room for improvement in this double lock-in strategy. So I'm just going to switch to a previous slide to explain that. So if we look at this spectrogram over here, the line width of this microwave peak is set by the electronic modulation of microwaves, and so it is a very tight line width. In comparison to this, we're normalizing this signal to the droplet peak, which has a wider line width of about 1 hertz, and that comes from small fluctuations in the droplet frequency. I think this can be improved further to have a better line width, and if this line width is on the order of magnitude of the microwave peak, then we would have even more sensitivity and better SNR with our definition of this dual locked-in contrast value. And so I think we would be able to access lower concentrations and see more dynamics with that.
Okay, yeah. So you basically need to improve the precision of the microfluidic control a little bit more, I see. And I guess what would happen, do you think, if you use like, this is maybe more instead of like a continuous wave laser, you use the pulsed laser, which then gives you a third like frequency modulation axis to normalize against. So you'd have like a triple lock-in. I don't know if that would work, but yeah.
I don't think that the sensitivity of this measurement by kind of applying more versions of lock-in within the same measurement will improve sensitivity drastically. Yeah, but I guess that's something to think more about. And what was the second half of your question? Sorry.
Oh, I think that was mostly it. I was just asking like if there was a consideration of using something like a pulse laser to, yeah, just to give you like maybe like triple lock-in. But I guess with the first lock-in, it was what, like 5% variation? I forgot what the number was on the slide,
15.
but yeah, so then the triple lock-in or the double lock-in got you to like 2%. So yeah, maybe it's
Mhm.
yeah, maybe another lock-in axis probably wouldn't get you much more. So I see. Yeah. Mhm. Yeah,
Yeah,
thank
I
you.
think that there are different strategies for the lock-in measurement. So we choose to use this droplet modulated flow because we're working with micro droplets, but you could do some sort of modulation just with an optical chopper or a pulsed laser. Um,
Mhm. Yeah, thank you.
Yeah. Now, please feel free to step in and ask your questions.
Hello, can I ask one question?
Please, student.
Hello. Thanks very much for the presentation. It's really interesting. I have two questions actually. One is I also checked the paper in Science Advances, but I'm really new in this area, so I just want to know what kind of spectrum do you detect? I mean, did you detect some typical peak for the single photon emission or even theta emission, or you just detect all of them? Is there any difference between them?
Yeah, so we're essentially we're using diamond particles that are 40 nanometers in size and each have like tiny ensembles of NV centers in them, and these particles are flowing through the field of view, so we're measuring some flux of NV centers, some steady state flux of NV centers.
So okay, so you detect the whole PL emission, or you just detect the typical wavelengths? Do you select the wavelengths, or?
Yes, yes. So we are detecting, essentially we use a long pass filter at around 700 nanometers and are collecting the red fluorescence.
Okay. Okay, I have another question then, because I when we look at your measurement like oscillation of this emission, that is has a very good contrast because of the you use you use microwave. Uh, what's the difference? Is it called a quantum emission? What's the difference if you directly detect the PL emission and I mean modulated with a pulse laser with a locking amplifier in the traditional way? What's the difference between the quantum setting and the traditional PL measurement setting?
Yeah, so I think we can call it quantum sensing because we are modulating the spin state of the NV center based on applying resonant microwaves. And so I guess your question is, can we do this without applying any microwave at all?
Yeah, actually, I guess we can do it just measure directly by PL measurement. Can we do that?
I see. So I think directly a direct PL measurement where we're just kind of modulating a laser and blinking these nanodiamonds, we would only kind of be able to measure a total brightness of the diamond, which would not give us information about the kind of paramagnetic ions inside of the droplet.
Okay, thank you.
And we have a question from Remy Blinder. Thanks for the interesting talk. Does release of protons from nanodiamond sources in the solution happen in a way that affects the value of the pH within the droplet? Is it known? I wonder if there is some ideal nanodiamond load for the mentioned applications.
Mm-hmm. So I think for these measurements, especially when we use gadolinium ions in the droplet, we also made a calibration curve where we add sodium chloride into the droplet to keep the salt concentration stable within the droplet. So that does not change our measurement. And I think while these diamond particles have a charged surface, we're not sensing some sort of pH change during these experiments. I think it would be interesting to see how different charges and pHs and certain loading concentrations of diamond in the droplet might optimize this experiment. But we haven't really measured changes in pH and how that affects the measurement.
And Adrisha, I also have a couple of questions. So one is, did you compare more closely these two techniques of detection of free radicals, the ODMR-microfluidics based and T1 relaxometry based? being developed in Ramana Shuragl group. So what is sensitivity, relative sensitivity of the two methods, and what are pluses and minuses microfluidic based technique in comparison to T1 relaxometry?
Mhm. So I think there's kind of two parts to this. The ODMR we're measuring is some indirect measurement of T1. The ODMR spectrum we measured does not really have much power broadening, but compared to maybe a stationary nanodiamond ODMR signal, there's possibly some broadening caused by this motion of particles moving through the field of view. And now to think about comparing this to T1. So possibly the benefits of this measurement are that we are able to do it with all this flow dynamics and integration of biological analytes. Implementing a T1 measurement with these flowing droplets and having kind of like a precise triggers for measuring along a T1 curve would be something that is experimentally quite difficult. And so by measuring contrast instead with this more simple measurement, we're able to do pretty well. I think it will be interesting, however, to implement a T1 measurement and really directly compare how good the ODMR is compared to T1. But it is not necessarily clear to me that this ODMR-based sensing is much, much worse than T1-based sensing.
And maybe second question. So again, with T1 direct sensing, there are a lot of experiment with intracellular sensing of some specific ROS production near specific organelles like mitochondria. So if to design similar experiments for microfluidic setup, say not with yeast cells, which are very difficult for nanodiamonds penetration through the cell membrane, but say with some animal cells. So what is the scenario for such measurements? You can just even speculate at the moment.
Yeah, so I think definitely for certain mammalian cells, uptake of nanodiamonds has been shown and is definitely possible. I think the picture of the droplet is interesting here in terms of selecting the number of diamond particles and interacting that with a single cell and then measuring multiple of these single cells. I think with kind of a small droplet profile, we could do some of these single cell measurements, and I could see essentially being able to possibly eventually do a flow cytometry like measurement where we can use some ROS sensing in droplet and have a flow cytometry like throughput of data for that.
And also you say on this slide you have a picture of lysed cells. Did you try yet yeast cell slicing, or it is just a possibility?
So cell lysing within microdroplets is a standard protocol used in microfluidics. And so we've kind of recently changed the aqueous environment of a droplet to have the correct kind of biological buffers to keep cells alive. And these buffers can be changed to cause cell lysis. So I do think it is a very real experiment that can be done. So far, we have integrated buffers that keep cells alive and loaded nanodiamond particles in the droplet. And lysing cells is just one step away from this.
Okay, thank you so much. Do we have any more questions? Okay, so Adrisha, thank you so much for your very exciting presentation. It's really great technology, and we wish you luck with further progress.
Yeah, thank you so much. All the discussions and materials that we get from you guys is key to these experiments, and so I'm excited to continue collaborating.
Okay, vice versa. Okay, thank you so much. Bye-bye.
Thanks.